One of the largest and most significant international scientific efforts in all of history, beginning in 1990 and ending in 2003, has been the Human Genome Project. It was a massive effort that began with discussions in 1984, and then through the actual sequencing by over 1,000 scientists across laboratories and research universities in 6 countries: the USA, UK, France, Germany, Japan, and China. Prior to the official publication of the results, the fruit fly genome was sequenced by 2000 and the mouse in 2002. These were 2 significant model organisms to be sequenced because of their application in research. Model organisms are non-human organisms that can be used in experimentation, and the research that is conducted with them can be applied or extended to humans. Model organisms are selected based on several criteria. They can be produced in large numbers through their reproduction. Fruit flies lay up to 500 eggs per reproductive event, and mice have between 3 to 14 pups per litter. There's a short generation time, meaning that organisms reach sexual maturity and can begin reproducing within a relatively short period of time. In fruit flies, it takes about 2.5 weeks from the laying of an egg until the fruit fly is sexually mature. In mice, the gestation period is around 3 weeks. Females then reach sexual maturity around 6 weeks, and males around 8 weeks. They are small and easy to maintain and care for. They are inexpensive to take care of. Most importantly, they share similarities genetically with humans. Humans and fruit flies share 75% of the same disease-causing genes. Humans and mice share between 95 to 98% of the same genome, including many disease-causing genes. Knowing the sequences of the genes of these organisms allow for researchers to understand the interactions between and of the inheritance of these genes. There are additional benefits too. Fruit flies are relatively simple to genetically modify or mutate, which gives researchers an increased understanding of the expression of genes. As for mice, since the genome is remarkably similar, they also have a similar anatomy and physiology, including the organization of the body into different systems. Mice can then be used to investigate complex diseases and to explain emergent properties that arise from the complex interactions between systems. Interestingly, mice genomes are easy to manipulate as well. It is possible for researchers to remove entire genes from their chromosomes to gain an awareness of gene functions. These specific techniques employed to sequence both of these organisms' genomes were different than the technique to sequence the human genome, and the techniques known have continued to change and improve since, allowing for further developments in scientific research. Some examples of the sequencing techniques used prior to and since the Human Genome Project include shotgun sequencing, double-barrel shotgun sequencing, Sanger sequencing, pyrosequencing, Illumina dye sequencing, SMRT sequencing, true single molecule sequencing, and larger scale sequencing. There are other techniques of sequencing for RNA, proteins, and polysaccharides. Regardless of the technique employed, thousands of genomes have been sequenced, giving us a better understanding of genetics and evolutionary relationships between organisms. Because there have been so many advancements and improvements in techniques, the human genome was sequenced ahead of schedule. After the publication, it became possible to sequence your own genome. It first cost $2.7 billion and 15 years to complete the Human Genome Project. In 2006, it cost $14 million, then $10 million in 2008, and currently between $1,000 to $2,000. The amount of time it takes to sequence the genome has significantly dropped too. With current technologies and techniques, it takes a little more than 1 day to complete. It should be noted that there are several ethical considerations to this process. First, should we know what genetic diseases we are susceptible to developing? Could this impact the way people select their partners for reproduction? And most importantly, could this become a basis for genetic discrimination? This was something of concern by scientists at the beginning of the Human Genome Project. They supported efforts of an ethical, legal, and social research program to address the many complex issues that might arise from this science. If you have not seen this movie already, check out Gattaca, released in 1997 in the midst of the Human Genome Project. It offers a pretty decent evaluation of the genetic discrimination and ethics with genomic sequencing, despite it being a dystopian sci-fi thriller. Since most, if not all of you, were born after this movie, please defer judgment. It justifiably has a certified fresh rating on Rotten Tomatoes. The technique employed in the Human Genome Project followed these basic steps. DNA was synthesized and broken into smaller fragments. From these small fragments, single-stranded copies were produced. Fluorescent nucleotide markers, a different color for each base, were used in the synthesis. Then the fragments were separated by length using gel electrophoresis to deduce the lengths of the fragments. A laser scanned the bands in the gel to cause fluorescence of the markers. An optical sensor could then detect the colors that fluoresced. The colors are deduced by a computer which translates the colors in the nucleotide base in each position of a DNA fragment. This technique has since been improved, allowing for more developments in research. Again, as time progresses, these newer techniques will be improved and offer greater insight into our own genetic domain.